ability to combine data sets together in this new way. Okay, so I know that I'm short on time, so I just kind of want to come back, though, to the idea of could we use tools like this to model complete organisms? So I think that it's an exciting thought and something that should motivate us all, whether you're interested in C. elegans or whether you're interested, in more complex organisms, to think about fully closing the loop, right? We all understand that the brain operates in the context of the body, which operates in the context of an environment, and we all understand that we're sort of impoverished in trying to understand the brain when we don't have those parts all put together. So I think that with a lot of the technologies that we've helped develop here, as well as a lot that's going on in other places, we're starting to get to the point where something like this might not be unimaginable, where we could actually collect together a lot of data, we could collect together a lot of structure, unify it together in three-dimensional environments like I'm talking about, apply physical simulations that have been used in many other realms of high-performance computing and physics, and start to get deeper understandings of how organisms work as a whole. So just to summarize, so I talked to you a little bit about the challenge of using modern information technology to understand and elucidate the complexity of the brain. I introduced Neuralex to you, the whole brain catalog. Didn't have time to talk about the multi-scale Connectome browser, but if you're interested in connectivity, I can talk to you a little more about that. I talked about this direct scientific use for the whole brain catalog in the olfactory system, and I talked to you a little bit about the possibility of using techniques like this and others that come in the future to create a fully data-driven simulation of a complete organism. And so with that, I'd like to acknowledge all these people who have played a huge role in putting this together, and I'd say thanks.
Do you know about the effort on the part of the Kaplan Institute and the Wellcome Trust through the Society for Neuroscience to create a kind of central database that seems very akin to what you're talking about?
I think so. If you say more about who's involved, I think I'll know even better. But the neuroinformatics community is pretty small. So with efforts like the INCF, I think we're all kind of 1 degree of separation, 1 or 2 degrees of separation away from each other. So that sounds familiar. There could be a few different ones that I know. One, which is a joint effort between University of Arizona and the University College London, is one that might be involved in that. But I think a few of these efforts now are coming together. The Allen Institute being an important node for that.
So you didn't really make it clear to me, at least, how you're going to verify information that's introduced in a wiki-style entry.
Okay.
What are you going to do there?
Yeah, so I mentioned just briefly about both aiming expert curators at the database as well as having a community curation model. So on the expert side, groups like the Neuroscience Information Framework and the International Neuroinformatics Coordinating Facility have connections into the academic community, and so they put together task forces of various kinds to both look at, how do we understand standards of naming brain regions, how do we understand standards of naming neurons, and through those task forces, they help to curate and maintain these shared databases. That's one. So that's sort of the expert model. The community model is one that has served Wikipedia at least fairly well, and so we're looking at whether we can really reuse that same thing, but the idea being that the more eyes that have a look, more knowledgeable eyes that have a look at a data set, and are able to make updates and corrections, the higher quality the data set gets. So in fact, when they look and compare, say, the Encyclopedia Britannica and the number of errors in that to Wikipedia and the number of errors in that, they actually find that Wikipedia has less errors. So both between specifically aiming experts at it and then a more casual aiming of the community at the data set, we believe that we can actually produce very high quality data sets as well. Maggie?
Could you talk a little bit about how when you're combining data sets,
How do you annotate the space? Okay, so good question. So a few things. So one is that the question of space and what are the appropriate spaces in the brain is one that you sort of have to hit brain by brain. So for the mouse, for example, the INCF has taken it upon themselves to say, we're going to have a single reference space they call the Waxholm space, and we can map all other brains to it, which means we can map the Allen brain to it, we can map brains that have been atlased by, say, George Paxinos and others. Through some transformational math, you can basically take any one of those different brain spaces and map them into a single space, so that when you're looking at cerebellum in your brain that you happen to be looking at is the same as the Allen brain, is the same as the Paxinos brain. So there's a whole effort in digital atlasing to make that possible. And essentially, you're reusing technologies that actually have already been applied to the geospatial world. So the same sorts of technologies that we use to understand where North America is, and look at North America either from a satellite or look at it from some geological survey. Those kinds of technologies are being applied here as well. So essentially, right, you can think of taking, once you have an origin and you build a coordinate system, right, you have a means of starting to make correspondences between spaces that may be somewhat different from each other. So 111 in my space and 111 in your space can be made the same through a simple transformation. Okay, so that's sort of the high level thing. What we try to do in our project is to make all that stuff extremely simple for someone who doesn't want to care about that kind of stuff. So the idea is that you, for example, put in an image into the whole brain catalog, and you can use tools that exist to just bring up a template of, say, the Allen space and warp your image into that space. So that's one way that user interface is an important piece of this. So, did I answer your question?
So then how do you account for
How do you account for individual differences? Yeah. So just real briefly. Right. So, for example, if you've got this cerebellum, which comes from an individual mouse that you happen to be looking at, you can use this tool here called Gibber, which was developed in the lab. Overlaid on top of it is a plate from the Allen Atlas. Okay, and by drawing little correspondences on this image, so you say this part of the brain is that, and this part of the brain is that, and this part of the brain is that. You can apply a warp to this image so that it can be fit together, okay, into just the right spot in the whole brain catalog. So that is one way where you can take a two-dimensional image. You know where the plate is because the plate is like the reference, right? And then you know where that plate is in three-dimensional space. And so you account for the individual differences by essentially taking your data and warping it and stretching it so that it fits into the generic space. And that's one way to do it. Maybe not the best way, but it's the way that we sort of started with. And then more advanced ways can be built on top of that. Justin.
It seems to me like you could also twist that. So the way that you just said that is you can fit your data into the generic space. Mm-hmm. But in a way, this also reflects we're talking about organizing facts, right? I mean, an ontology is just a philosophy of what is what, mm-hmm. right? And so by warping individual differences into a generic space, you're essentially consolidating and saying that there is a right space and that everything else must fit into that space. And I think that there are other types of individual differences that that one might see, and it's actually in those individual differences and in the variability that I think that we find some really interesting things as far as research goes. How do you rectify that? I mean, do you keep track of what the warping parameters were of an individual of a piece to see, like, to say, okay, because it could be really interesting to, if you have a big enough data set to say, all right, so like overall, we've had 15,000 submissions of this, like one part of this 15,000 submissions of slices of the cerebellum. What are the parts that tend to be warped the most relative to other parts? Mm-hmm. Do you guys, to what extent can this kind of a framework keep track of that kind of variability and the interesting scientific questions that one could ask with that approach?
Mm-hmm. Yeah, great question. Actually, I think there's a sort of a set of questions in there. So I'd answer it in a few ways. One, yes, you can save the warp. So the nice thing about the warp is that it's a mathematical structure. It's something that's very well defined, and so that can be attached to this data so that you could go and study that. I would say that there isn't anyone yet who's really digging into what I think you point out is a really meaty area of looking at individual differences, but part of the idea of publishing all this data and exposing it and making it available online is so that folks can go and answer those kinds of questions or even start to approach those kinds of questions, because until you have that data, you can't mine it, right? And the last thing, I guess, on correct. So this there may not be a single correct space. What we try to get at is a space that is useful for being able to pull together data into a way that we can then do some kind of analysis. So with the Kristen Baldwin example, for example, she was looking at multiple individual mice and they all maybe had their individual differences. We brought them together in a space to try and start to at least ask the question of how do these neuronal morphologies look the same or look different across individuals. And so what's at least the least that you can say about how they come together. And then using math, you can pull apart what are the individual differences a little bit better. But until you have that, a little bit of rigor, right, you can't even approach those questions. So, so I think that's extremely valid, extremely important, I would love to talk to you more about it. Good question.
Can we come to the talk? So I would start out by saying, know how long your designated time is and fit the talk to that time. We have a lot of interesting information that you glossed over simply because you didn't know how long to plan your talk.
Mhm.
Drag.
That's good. Thanks.
I saw your talk a couple times, and each time I see it evolving, and I think this is like 1,000 times better than the first one, and I thought the first one was really good.